Well, it's a pleasure today to introduce Barbara Niethammer, who will speak in the seminar in the analysis of PDEs. We're overjoyed to have her, and she's going to speak today on an obstacle problem for cell polarization. Thank you, Barbara.
Yeah, thank you very much for the kind introduction and the invitation. It's an honor to speak about our work here on the obstacle problem for cell polarization. And this is joint work with my colleague, Juan Velazquez, and Matthias Roeger from Dortmund, and our PhD student, Anna Logioti. So first of all, what is cell polarization? So this is a key ingredient for several cell functions, and in particular for the migration of cells. And it's characterized by a large spatial variation of the concentration of certain proteins on the cell. And here I took some pictures. This is from a review article by Appel and Edelstein-Keschet. And here, for example, in picture A, you see a cell that's actually moving to the right. And it has a high concentration of actin in the front here. So that's indicated by the red color. And a high concentration of myosin at the rear. That's indicated by the green color. And somehow due to this polarization of these proteins, the cell is able to move. And it's also indicated by this cartoon down here. And there are actually several reasons for polarization. And in the following, we are interested in polarization due to an external chemical signal. And the goal is to understand in a simple or relatively simple mathematical model how and when polarization occurs. And here's now the setting for our mathematical model. So the cell is just represented by a three-dimensional domain omega with a smooth boundary. The boundary is called gamma and represents the cell membrane. And then we have two versions of a protein on the membrane indicated by this blue and red color. And the concentration of the active version of the protein is denoted by U. And there's an inactive version of the protein, and the concentration is denoted by V. So U and V are non-negative functions that live on gamma. But then the inactive version of the protein can also move to the interior of the cell, and the respective concentration is called W. So W is a function that lives in omega and is also non-negative, of course. And as I already mentioned, so we are interested in the effect of an external signal, and we assume that this is already processed and represented by a density function f. So f is, we assume it's strictly positive, continuous, and so also bounded on gamma. And what we want to do is now in the first step is to set up a bulk surface reaction diffusion model with two main ingredients. So one is activation by the external signal, and deactivation by what is called a Michaelis-Menten law. So what does that mean in formula? So that's here. So this is an adaptation of a model which has been suggested by Andreas Retz and Matthias Roeger in a slightly different setting, but adapted here to our situation. So first of all, we have an equation for the density of the active protein U on gamma. So here we have diffusion on gamma, and I should say here with some abuse of notation, we also call the surface Laplacian delta. And later we have only surface Laplacian, so there's hopefully no reason for confusion. We also put already here the diffusion constant to be 1. And then we have an activation term, which we just assume to be proportional to the product of the density of the signal F and the density of the inactive protein V, and we have a rate constant R. And then we have deactivation by, that's what I mentioned, is called the Michaelis-Menten law. So that's this nonlinear function. The precise form is not so important. It's just it's strictly increasing, bounded, and 0 at 0. And then there is another rate constant S. Then for the inactive version of the protein, we have similarly, we have diffusion. Diffusion constant is the same as for U, so we set it to 1. Then we have activation and deactivation term, but with the opposite sign, of course. But then we also have that the inactive protein can move to the interior of the cell, and this is described by these two exchange terms. And then the equation for the inactive protein within the cell is just pure diffusion, but here we have a diffusion constant D, and typically this D is much larger than the diffusion constant on the membrane. So since we set the letter 1 to 1, so we assume D is typically much larger than 1. And then we have this Robin boundary condition, which describes the exchange between the membrane and omega. Yeah, we could, of course, incorporate additional mechanism, and we have done that in the paper, but for the purpose of this talk, I want to keep it as simple as possible, but still see some interesting phenomena to show up. Okay, so very briefly, a comment on well-posedness for this reaction diffusion model. So there is a result by Hausberg and Roeger for a related model, but the proof can be easily adapted to obtain existence and uniqueness of a non-negative weak solution for all times. And the important property is that this solution conserves the mass. So the total mass is just the integral of W over omega, so W concentration of inactive protein within the cell, plus the integral over gamma of U plus V. So the sum of these integrals is always equal to some mass M for all positive times. Okay, and in the first part of the talk, I will mainly discuss, in the first part, I will only discuss steady states. So I will consider certain scaling limits of steady states. And first of all, we remark that from the existence results, theorem 1, by some fixed point argument, we can also conclude on the existence of a steady state. So for any mass M, there exists a non-negative stationary solution with mass M, and by simple regularity theory, we also get that this is smooth. So the steady state then is a solution of this system of equation, which is just the same as before without the time derivatives. Yeah, and as I already mentioned, I will discuss several scaling limits of this system. And the first one we will perform is to consider the limit of cytosolic diffusion coefficient D going to infinity. As I already mentioned, so in typical application, D is much larger than 1. So it's a reasonable thing to let it go to infinity first. And if we let go D to infinity, this forces W to be constant, so it's not depending on space anymore. So W will just be a constant. This constant still enters here and is a kind of Lagrange multiplier, which ensures that the total mass is conserved. And it's not difficult to perform this rigorously. I just list the result here. Yeah? So for D to infinity, so we consider a sequence D to infinity, the corresponding solutions converge to solutions U and V and W of this system here, where U and V satisfy the same reaction diffusion system as before, but just W is a constant, as I said, and it's determined by the constraint that the mass is conserved. And here we, without loss of generality, the measure of omega is 1, otherwise we would have another constant here. Okay, so far so good. So here's the steady state version of the reaction diffusion system for D to infinity. And now I can come to kind of the main part of the talk, which relates to this issue of polarization. So the key issue in this polarization is that you observe that relatively weak chemical gradients still lead to large variation of the concentration of the proteins on the membrane. And the reason for that is that you have large rate constants in the system. And to explore this in our model, is what we want to do now is to go over to the limit of large reaction rates. So what does it mean? So we replace these rate constant that I call A, B, R, and S. We rescale them by a small parameter epsilon. And then we also consider we need to rescale the mass by 1 over epsilon. So we look at large masses. And if you plug this into the equation, then you also see that relevant terms only balance if also U the concentration of the active protein is of order 1 over epsilon. So to get something later of order 1, we introduced a new function. U epsilon is defined as epsilon times u. And it's important to remember this rescaling later on, because what we're going to do is to pass to the limit epsilon to 0. We get a new problem. This is actually will be the obstacle problem that appears in the title of the talk. And for this limit equation, we will say that we have a polarized state if the solution U is 0 in some regions of the membrane and positive in other regions of the membrane. So in these original variables, it means that that U is very large in some regions and small in other regions. Okay, so here I just wrote the system of equations again. I just after rescaling, I put for simplicity A, B, R, and S equal to 1. U epsilon is the rescaled U, but V and W remain unscaled. And then you get this system of equation, and because you don't rescale V and W, you have an epsilon in front here and here. Okay, and so now we want to pass to the limit as epsilon goes to 0 in this system. This is not very difficult, but let me somehow indicate it's not really a proof, but somehow the main steps, just to see where the limit equation comes about. So here I just copied again the system that we saw before. Okay, so first of all, we already know U epsilon, V epsilon are non-negative functions, and W epsilon, remember, is just non-negative numbers. And one nice thing is if you integrate the equation over gamma, then the Laplace terms drop out because we have a closed surface. And by integrating and combining the terms, you easily get first uniform L1 bound on U epsilon and V epsilon gamma, and a bound on the numbers W epsilon. And so you have the first compactness result for subsequence. U epsilon converges weakly to U and V epsilon to V in the sense of measures. And the sequence of numbers W epsilon converges to a number. And I'm sorry for that, but we used to call it alpha. I'm so used to alpha, so I call it alpha now because otherwise I get confused. So alpha is the limit of the W epsilons. Another simple bound you get is that this Michaelis-Menten term is, of course, bounded by 1. It's non-negative. So you get a weak star limit in L infinity that we call psi. Psi, of course, depends in some way on the limit of U. We don't know yet how exactly. But then we look at the equation for U here, and you see, okay, we already said this term converges to this psi. The Laplacian of U in the sense of distribution converges to Laplace of U. So it remains to identify F times V epsilon. And this you get from the second equation, because in the limit, the Laplace term drops out. And this here converges to psi, this converges to alpha. So you get in the limit F plus 1V is alpha plus psi. And then you plug this into the equation here. And then you get the following limit equation for U. And here I introduced a new function just for convenience. As if you see here's the F plus 1, you multiply with F. So you typically get an F over F plus 1, and we call this function G now. So G is now our external signal, and it's bounded by 1 and it's non-negative. Okay, then so we get this equation minus Laplace U is G times alpha minus 1 minus G psi. So the minus psi comes from that term, and the G alpha plus psi comes from this term. But still, I mean, alpha is a constant. Again, it's a Lagrange multiplier to ensure the mass constraint that we also have now for U, because here in the limit, this term and this term drop out. So in the limit, we have the integral of U is equal to M. But we don't know yet really the relation between this function psi and U. But what we can do is to do a better convergence, to get a stronger convergence result. We also get that U epsilon converges to U in L1. So pointwise almost everywhere. And this means if you just remember the form of the Michaelis-Menten term, that psi is 1 whenever u is positive. Or in other words, psi times u is equal to u almost everywhere. Okay, so this was maybe a bit messy, sorry about that, but let's summarize the result here. So in the limit, the steady state u epsilon converge in L1 to some non-negative L1 function u. The Michaelis-Menten term converges weakly star to some function xi, but xi must be 1 in the set where u is positive. And there exists a non-negative number alpha such that u solves this elliptic equation on gamma, and the integral of u is equal to a prescribed mass m. And here's now finally the obstacle problem that appeared in the title. Why obstacle problem? Well, you see, if in the region where u is positive, u solves a nice elliptic equation. Here xi is 1. But then there are also regions on gamma where u is just 0. Or potentially there's a region where u is 0. Okay, and I should also mention that we have some further regularity on u, so u is quite smooth. It's actually in W2p for all p less than infinity. So, okay, so this is now this equation that we want to study a bit more. So remember what we are interested in. We are interested in polarization. And I mentioned this already. We say we have a polarized state if the solution of this problem if both the sets where u is positive and where u is 0 have positive measure. And in principle, we expect that for large m, for large mass, u is strictly positive. Then we would not have a polarized state. So what would we like to know? I mean, we would like to know that for given m, that there exists a unique solution to our problem. We want to identify a range of masses m for which we have a polarized state. So we expect for large m, we do not have polarization, but we want to identify a critical value up to which we have that. And ideally, we want to say something about the polarization patterns. Now, how to approach this? Well, so first of all, there's simple observation. So first of all, we observe that the solution u is constant if and only if g is the external signal is constant. So this is of course boring. So we assume from now on that g is not constant. So the next observation is that this alpha here, the Lagrange multiplier, cannot be arbitrarily large. Because if you integrate I put here the equation for you again, if you integrate this over gamma, then you actually get that alpha is just this integral over 1 minus g times xi divided by an integral of g. And since xi is bounded by 1, you get that alpha must be smaller than a number alpha star, which is just determined by the data. So we have immediately a bound on the possible values of alpha. And then another important value of alpha that will appear in a minute is this term here is bounded below by the term when you replace g by the maximum of g. So this I call alpha naught. And since g is not constant, alpha naught is smaller than alpha star. And you will see the interesting interval for alpha is just alpha naught, alpha star. Okay, so with this observation, so we now proceed, and we take now a slightly different point of view. We fix alpha for a moment and then solve the obstacle problem for fixed alpha and then see what masses we get. And the advantage is that if we fix alpha, and we can formulate the obstacle problem as a variational problem. And so we fix alpha and then we minimize the corresponding energy. So this is just the Dirichlet energy. This corresponds to the right-hand side when we put psi equal to 1. And we minimize this energy over all functions that are non-negative, all H1 functions. And then minimizers correspond to solutions to the obstacle problem for fixed alpha. And then what you get is immediately by the direct method of calculus of variation that for all alpha between 0 and alpha star, a minimizer exists. And actually, you also see here what goes wrong if alpha is larger than alpha star, because if alpha is larger than alpha star, you can just take V to be a constant n, and then this goes to minus infinity, so the energy is not bounded. Okay, so for all alpha smaller than alpha equal alpha star, minimizer exists. What you also see, if alpha is smaller than alpha naught, then this term is also, this is always non-negative, so our minimizer is just 0. So this is what I mean with that the interval alpha naught alpha star is the interesting interval for our alphas. Then what we also get by some monotonicity, that minimizers are unique for alpha smaller than alpha star, and I forgot to write it here for alpha larger than alpha naught, of course. And they are not unique for alpha equal to alpha star, but alpha equal to alpha star, you can always add an additive constant. And finally, we also have a very strong, sometimes very strong monotonicity. So the map alpha to the solution u psi is strictly increasing on the interval alpha naught alpha star. So this also means by increasing alpha, we can increase the corresponding mass m. Okay, and what you also see already here, that this value alpha star is somehow crucial to identify the critical mass m star. Remember, I want to find a critical mass below which we have polarization and above which we have no polarization. So why is this alpha star important? Because assume you have a minimizer for alpha equal to alpha star. Assume that's a polarized state, but then you can just add a constant and it's not polarized anymore. So this shows somehow already that alpha star is important. And in fact, we can use this alpha star to define the critical mass m star in the following way. We put alpha to alpha star and psi to 1 in this, we have this simple elliptic problem. This solution is only determined up to a constant. And we fix this constant by setting the minimum of u star to 0. And then we define m star to be the integral of this u star. And then we can show the following. So for any mass m, there exists a unique solution u alpha, and in principle also psi of our obstacle problem. If m is larger than this critical value m star, then alpha is alpha star. And we have no polarization, so the zero set of u has zero measure. And the solution is just given by this u star that we constructed up here. And we add a constant to fix the mass to be m. Then if m is smaller than m star, then we have a polarized state, a set where u is 0 has positive measure, and alpha must be smaller than alpha star. And if m is equal to m star, then polarization may or may not occur. And that depends on the details of the signal G and perhaps also on the shape of gamma. And somehow we have indicated this theorem in this picture here. You see you have this monotone relation between m and alpha. If m goes from 0 to m star, alpha varies monotonically from alpha naught to alpha star. And if m is larger than m star, then alpha is just constant to be alpha star. Okay, so this answers our first question. So given a mass m, there exists a unique solution of our obstacle problem, and we identify critical mass m star, and it's even constructive. We give a formula for this m star. Okay, so then another question we had, so can we say something more about the shape of u depending on the external signal G? And so here is one result in that direction, which we call localization. So what we expect is that the concentration of the active protein U becomes large where G is large. And here we have a result in that direction in the limit of small masses. So with S, we denote the points on gamma where G, the external signal, attains the maximum value. So here in this picture, this is just this point. And then we consider a sequence of masses, Mn, that go to 0. Then the corresponding solution of the obstacle problem Un converge to 0, but we have a localization in the following sense. if we take a delta neighborhood, so here let's say this is of order delta, of this set S, where G is maximal, then for any positive delta, there exists a knot such that UN is 0 outside the neighborhood for all sufficiently large n. Okay, so this kind of confirms this expectation that U concentrates where the external signal is maximal. Okay, so where are we now? So I described now the results for the steady states for this obstacle problem. And now I want to say something about the parabolic problem. And recall that the starting point was this reaction diffusion system. And we then looked at steady states, then let t to infinity and then epsilon to 0. But we can also start from the full reaction diffusion problem, let d to infinity and then epsilon to 0 without going to steady states. And what we get is the analogous parabolic obstacle problem that I wrote down here. It's basically, I mean, it's this, just instead of Laplace u equal to the right-hand side, we have now the dtu minus Laplace u is the same right-hand side as before. U is non-negative. Psi is 1 where u is positive. We have initial data, and the difference, of course, is now alpha is not just a number, it's a time-dependent function. And you can get different representations of alpha, either by integrating, we saw that before, by integrating this equation over gamma, then the left-hand side drops out. We get alpha of t is the integral of 1 minus g times psi, divided by the integral of g. So alpha depends in some way on u through this function psi, or you can just integrate over the positivity set of u, then you get this other representation of alpha. Yeah? It's the integral of 1 minus g over the positivity set of u divided by the integral of g over the positivity set of u. So you see somehow this one unusual kind of relation between alpha and the support of the solution u. Ah, yeah, and then I forgot, yeah, and also for this system we have conservation of mass, so the integral of u is equal to m for all times. Okay, so we can, as I said, you can get solutions to this problem by a similar procedure, by letting epsilon to 0 in the reaction diffusion system. But what about now uniqueness of solutions and the long time behavior of solutions? Do solutions of this obstacle problem converge to steady states? And then it turned out, and it was quite a bit unexpected for us, that this evolution is very nice in the sense that we get an L1 contraction. What I mean is the following. So we have U1 and U2, so two solutions and corresponding alpha 1 and alpha 2 and psi 1 and psi 2. And then you look at the positive part of the difference of two solutions, integrate over gamma, and then this functional is decreasing in time. And as an immediate corollary, you get uniqueness of solutions. And with a little more work, if the external signal is time independent, then steady states are globally stable. So in fact, you for given mass m, the solution converges as time goes to infinity to the steady state with the same mass. And actually this L1 contraction property, so this relies on a quite delicate balance of the non-local terms in the equation. And just to get you an idea, I give you a glimpse of the proof of how this works. Okay, so this is just a little technical, but okay, let's see. Yeah, so we look at two solutions, U1, U2. We look at the positive part of U1 minus U2. Chi plus is the characteristic function of the set where U1 is larger, U2. Okay, we differentiate and see what we get. So the Laplace term, you can easily see that the term coming from the Laplace is not positive, so we drop it here immediately. And then we get two terms which involve the difference of the psi and the difference of these non-local functions alpha. And now what I want to do is next to, you need to replace this somehow rewrite this difference of the alphas in terms of the xi's. And for this, we just recall that that alpha times integral of g is integral of 1 minus p psi. And this I just used here to get this. And then we split this integral here in the set where at least one of the u's is positive. This is indicated by this new function chi, and the set where both functions are 0. This is the set where 1 minus xi is 1. Okay, and then we use that in the set where both functions are positive, we have these pointwise relations between alphas and xi's. And this we plug in here for this, this already happened here, from here to here. We plug this in for 1 minus t times chi 1 minus chi 2, we get this in. We get this alpha 1 minus alpha 2 and combine it with this term here. And then get a relation of the difference of alpha 1 minus alpha 2 with the xi 1s and xi 2. And then we just, okay, sorry. As you see here, the denominator, we multiply the equation through and plug it in, and then get that the time this quantity, which we want to have non-positive, times this number. We plugged here, just plugged in what I got from the previous slide, and then we rearrange a bit, and then we see we get two nice terms. So chi plus is the region where u1 is larger than u2, but then there's also xi1 is larger than xi2, so this is also larger than or equal to 0. This is larger than or equal to 0 with the minus, this is smaller than or equal to 0. And a bit similarly here we have the set where u2 is larger than u1, but then xi2 is larger than xi1. It's also non-negative, and with the minus here we have in total this is smaller than or equal to 0. Okay, so this was maybe a bit in a sense boring, but it as I said, we somehow did not expect it, and we're kind of a very nice surprise when we figured out that this works. On the other hand, you also see from the proof that it's probably impossible to get a kind of rate of convergence to equilibrium or anything like that. Okay, yeah, then what I told you so far was all about the case that we let in the beginning, the cytosolic diffusion coefficient d going to infinity. And let me tell you now a little bit about the case when we don't do this, and we keep d to be finite. So that means, what I wrote down here is the full bulk surface reaction diffusion system, but we have already scaled the rate constant and u by this parameter epsilon, and we also actually need to rescale d by 1 over epsilon. And then you get this system of equation here, and again we want to let epsilon go to infinity. And in principle, the first two equations, that is the same as before, yeah. I mean, just that you keep the dtu here, Laplace u. This becomes this function psi, and f times the epsilon, the limit we get from the second equation. And the difference is just that w epsilon now converges in the limit to some function w, which is not constant in space anymore, but rather solves an elliptic equation, because I mean this term drops out in the limit and the rest remains. Okay, so and then we obtain this non-local obstacle problem, which, okay, so the equation for u looks as before, just instead of alpha, that was a function that depends only on time, we have now w that depends also on x. So psi times u is u, almost everywhere remains the same, but w now solves this equation. It's harmonic inside omega and satisfies this Robin type boundary condition. Yeah? So this is, okay, I call it a non-local obstacle problem. The other one was also non-local in some way. This is a bit more non-local if you want. Because for each time t, you need to solve this elliptic problem, and then the solution goes in here. Okay, so this looks now much more complicated than the problem that we had in the case d to infinity. But it turns out that we can get exactly the same results. I mean, we get existence and uniqueness of steady states for given mass m. We can construct a critical mass for polarization of the steady states. The construction is a bit more complicated, but we can do this. And for the parabolic problem, it's also an L1 contraction. So we get uniqueness of solutions and global stability of steady states. So exactly the same results as for the case d equal infinity. Okay, and yeah, but I will spare you any technical details of that. That's just all I wanted to say about the case d smaller infinity. And okay, so these were kind of very nice results, we thought, and then we wanted to move on and to study maybe a bit more qualitative properties of solutions. And we also thought, motivated by some discussion with the biologists, to think about a fast changing external signal and maybe do some homogenization in time or something like that. But when we started thinking about it, we realized we would like to have a bit more regularity of the solution. And then we jumped into this issue, and that turned out to be a nuisance. So with regularity, what I mean is regularity in time. So here I wrote down the obstacle problem, again for the case d equal infinity. That's for this thing, this is difficult enough. And remember, we have this non-local function alpha with this kind of dependence on the support of u. And a priori, alpha is just in L infinity in time. And the question is now, it would be very nice to have alpha continuous and to have more or less equivalently that the support of u changes continuously in time. And it's kind of clear that in general we cannot expect it. Why? Because if we take initial data for which this right-hand side is positive, then by the strong maximum principle, solution will be positive for all positive times. That means the support of u will jump in time, at least initially. And then, what we try to prove is, okay, if we assume our initial data are such that this is smaller than 0 or even bounded away from 0, can we then prove that the solution is at least continuous? I mean, alpha is continuous as t goes to 0 and also the support of u. And we could do this. So here is some indication of the result. So this is what we have on the right-hand side at time 0. If we assume that this is bounded away from 0 in the set where the initial data are 0, and we have some regularity on the boundary of the support of the initial data, then in fact we have continuity of this function alpha and of the support as time goes to 0. However, if this condition is not satisfied, then as we already expected, then in general alpha will jump at 0 and also the positivity set of u jumps. And we have a kind of characterization of these. We can even characterize these jumps. But of course, you might ask, okay, this seems to be, I mean, if the condition is not satisfied, is an incompatibility of the initial data. But what happens for later times? And unfortunately, we don't know. I mean, we have some numerical simulations which might indicate that even later jumps can happen. But okay, we simply don't know. But what we did is we looked at some specific one-dimensional data. So we assume we take initial data, they are axisymmetric, and in the one direction in which they change, they are monotone. And then this property is pertained for the data as positive times. And for this particular case, we can prove global continuity in time. But in general, we don't know. Okay, and I realize I was a bit faster than expected. So, but here's a summary and outlook. So we proposed the bulk surface reaction diffusion model and following the work by Matthias, Roeger and others for cell polarization. And we look at a large reaction rate limit and obtain different versions of obstacle type problems. And for those, we could we could find a critical mass, which defines polarization of steady states. We also have some results on the localization. So the concentration of the protein becomes large where the external signal is large. For the parabolic problem, we have global stability of steady states, and we did a bit of regularity theory, which is really delicate, and there's maybe more to be done, but yeah, okay. And as I said, the outlook is to understand a bit more about properties of solutions. So for example, we want to understand what happens if we have a fast changing external signal. Okay, then, okay, here's some papers also, and I thank you very much for your attention.
Thank you. Thank you very much. Very nice talk. Please, questions from the audience.
Felix has his hand raised.
Okay.
Okay, so can I just talk?
Yes, please do.
Very nice talk, Barbara. So I had the following question. So before you kind of when you were still considering the case of infinite diffusion, and you were looking at the stationary states, and you were looking at concentration of U, your finding was that U concentrated at the maxima of G, right?
Yes.
Yes, okay. Now, so it's in a certain sense the inhomogeneity of G that triggers the pattern of U. Now, my question is the following: if you consider the case where D is finite, could it be that even at constant G, you would see inhomogeneities because of curvature effects?
Yes, I mean, that could be. I mean, this is another interesting question that I did not mention, which the curvature of G, of gamma, you mean?
The curvature of gamma, exactly.
Of gamma, yeah, that should play a role, and it would be very nice to explore that, but I mean, we haven't done
because
it.
Then in a sense, I mean, since this polarization is, you know, probably changes the shape of the cell, there could be some self-reinforcement of this.
Yes, that could also be. Okay, this would be another thing to really couple, you know, say the evolution of, to let the membrane also evolve. Or
yeah, or at least see that there might be a kind of some reinforcement of that, some positive reinforcement that the cell becomes polarized, because when it's already a little bit ellipsoid, that will, you know, go in that direction.
No, that would be very nice. I mean, we haven't done anything yet in that direction.
Thank you.
Another question. Please go ahead and I can't, for some reason, I cannot see your raised hand, but if you have a question, please go ahead and simply ask. Let's check here. Now I can still, let's see here. Make sure I have, okay, let's see. I have a question. Actually following up on Felix, in the sense that, you know, what you've said is kind of, it generates a fountain of questions. And in particular, when your D, in the bulk is finite, one may be able to expect some surface to volume ratio effect of the cell. So again, cell shape could appear,
yeah,
you know, because of the non-local problem.
Yes, yeah. Unfortunately, we didn't do anything in that direction yet. So hopefully we can come to that as well. But at the moment, can't say more, I'm afraid.
I can ask another question. So of course, I'm pleased to see an L1 contraction principle there. So what is seen from the original system? What's the effect which destroys the L1 contraction principle? I mean, so what? I mean, I think you were right to be surprised. I would have been as well to see a L1 contraction emerging here. So what? At which place do you gain it in this singular limit?
Actually, for this system, you mean the original reaction diffusion? This one, no, sorry, this one is also an L1 contraction, I think. But I mean, the thing was that we started with more terms in this model, which for the purpose of this talk I threw out. With these additional terms, there's definitely no L1 contraction.
Okay.
So, but in the large reaction limit that we looked at, these other terms, yeah, what can I say? I mean, this is also an L1 contraction. So in some
okay,
sense,
so
but we didn't look at it because we had more terms. We only realized it on the level of the obstacle problem. So, yeah, maybe this is disappointing that way. No miracle appearing.
Okay.
Again, if anyone has a question, please speak up. For some reason, I can't see a raised hand, so. Well, I'll again take the liberty, since this really does raise many questions. You know, again on taxis, for example, you know, movement of cells in bulk. Have you any thoughts on you know, how this polarization and the associated motion of cells in a signal would flocking or clumping or anything?
Say again, I somehow I don't understand very well. The audio is not.
Oh, just would you this model that you have analyzed, would it suggest some sort of bulk motion of cells? Could you understand some sort of, given a signal, cells would move?
Okay. Yeah, I mean, this is all something which one should explore further. Maybe I can tell you what I said. We started on this after discussion with a biologist from Bonn, but somehow we then dived into these mathematical questions, and we have to get back to him. But from what I remember, the main question was, I mean, you have these signals, so the cell is moving, say, to the right, and then you change the signal, and then it moves in the other direction. But the question is, how does it really move around and goes, or does the polarization just change and then it just moves in, so head and tail exchange in some sense? And this was one of the questions for them, when this happens and how. And this would be very nice, but of course then to figure this out in a mathematical model, but of course then we have to extend it quite a bit, let it move. But this was the reason why we wanted to understand the fast changing signal. So can this polarization follow immediately, or what's going to happen then? That was the idea, but somehow, yeah, haven't got to that yet.
Thank you. Well, thank you very much for a very nice talk and introducing a novel problem and outstanding analysis.